Hello. This is the recorded lecture for Chapter 24 for Anatomy and Physiology 230. And in this particular chapter, we're going to look at the urinary system, and we'll look at the architecture, obviously, as well as the physiology of the urinary system. We'll talk a lot about how the renal system goes through the filtering process, which is kind of poorly understood by most people because they don't really understand how that function works. And so we'll talk through that a little bit and sort of hopefully we'll clarify that. In the urinary system, the kidneys are the major excretory organs. They're the major organs of filtering, as it were. And the bladder is the storage reservoir for our urine. The tubes that run from the kidneys to the bladder are called ureters, and the tube that runs from the bladder out to the world, so to speak, is the urethra. And so we see that as our basic organs and how they're put together. So we have a kidney right and left. We have ureters that extend from the renal pelvis. That's the sort of the inside curvature of these little bean-shaped structures. Bean-shaped structures, not little. The ureter, which runs down and will insert in the bladder, actually posteriorly and inferiorly on either side. The bladder is located here, and then the tube that runs from the bladder to our outside world becomes the urethra. Now you can see that the arteries that feed arteries and veins that feed each of the kidneys is called the renal artery and renal vein, respectively. There's also a lymphatic system that feeds us also. You can see also that the right kidney is a little lower than the left kidney, and that's because there's a big liver up here that sort of puts pressure and pushes that down a bit. You can also see, just for for complete purposes, you'll see these little top hats, if you will, that sit on top of each kidney, and those are the adrenal glands. Now, the function of the kidney, obviously, is to remove toxins and metabolic waste and excess ions from our blood, but it also helps regulate blood volume, our chemical composition, and it controls the level of free hydrogen in our body. We call that pH, or pH, which measures really our levels of free hydrogen, and that's what it manages. Kidneys also function in gluconeogenesis. Remember, neo means different or from a different source. In this case, it's producing glucose from a non-carbohydrate source. In this case, maybe protein or fat. So it's responsible for gluconeogenesis during prolonged fasting. And its endocrine functions include the production of renin. Remember the renin-angiotensin system, its regulation of blood pressure and kidney function, where the renin creates angiotensin, and we'll talk about that again. And then we talked about this before. We talked about blood, erythropoietin, EPO, which is a hormone secreted by the kidneys that manages red blood cell production or can increase red blood cell production. It's also the location for activation of vitamin D in our bodies. Now, the kidney's anatomy is they are retroperitoneal. As you remember from the digestive system, retroperitoneal means behind the peritoneum. And they're located in the superior lumbar region, posteriorly. We know the right kidney is lower than the left, as I said earlier, because of the liver. And they're shaped kind of like a bean. They have a convex lateral surface and a concave medial surface. And the medial surface is often called the hilum. So the renal hilum leads to the renal sinus, and we'll talk more about that in a bit. The ureters, the blood vessels, lymphatics, and the nerves all enter the kidneys at that hilar area in the hilum. Now we talk about the layers of the kidneys. We talk about the outside-in layers, as it were. The renal fascia is the anchoring outer layer that this fibrous connective tissue that kind of holds the kidneys in place. And there's also fat around the kidneys, which serves as a block for injury or that kind of thing, gives us some protection. With last of all, there's a fibrous capsule around the kidneys, and it's a very dense, greedy kind of capsule. If you were to take a scalpel and try to cut through that, it's kind of a greedy sort of substance. And that helps prevent the spread of infection to the kidney from the rest of the body. As we look at this transverse cut, we're going through the kidneys and basically the hilum. You can see there's the hilum on each side, if you will. And we see the peritoneum, which is in front of the kidneys, so that makes them retroperitoneal. And they sit just really to the right and left, if you will, of the lumbar lumbar vertebrae. In this case, L2 is really where the hilum are usually at, the lumbar 2 vertebrae. What you're seeing is you see that you see the renal fascia, you can see that, the anterior posterior perirenal fat capsule and the fibrous capsule. You can see those layers as we go in, and you can see that there's that fat that surrounds them on purpose to give us that protection. It's there on purpose. As far as the internal anatomy of the kidney, so from the kidney source in, there's the renal cortex, and that's that granular superficial covering around the kidney. When I say granular, like I said, it feels like sandpaper, very light sandpaper. As we go in for the cortex, there's the renal medulla. That's kind of the center of almost everything we hear medulla. And it's made up of these cone-shaped pyramids. They're called medullary pyramids or renal pyramids. And they're separated by these little columns. And we'll hear about lobes, and that's a medullary pyramid. It's a medullary pyramid and surrounding cortical tissue. And you see the difference in those two. There's a papillae, that's the tip of the pyramid where the urine actually comes out, and the renal pelvis, which is the funnel-shaped tube, it ultimately goes to the renal sinus. You have major calyces, the urine flows from pelvis to the ureter. And let's talk about this a little bit. So here's our cortex. Here's our outside cortex. And there's our outside cortex, and inside we see is the medulla is really this part right here, and you see these little upside down pyramids, or these pyramids here, that have the points that are pointing towards where urine is collected. So this is where the urine is filtered in these areas, the cortex, the medulla. You see the columns in between each of these pyramids, and so there's a renal pyramid, and it comes to the point which is called the papillae of that, and it's this point where urine, whatever comes out of there is actually urine. And it's collected in these small areas called the minor calyces, and the calyces are just collecting areas, and they feed to a major area called the major calyces, which feeds to the renal pelvis, and that's where all the urine eventually runs to until it goes to the ureter, begins going down the ureter. So you get an idea of what we're talking about. So there's the cortex, and this is the medulla made up of those pyramids. I have a papillae at the point, and the columns around each side. So that's kind of the feeding system. Now, if we look at this on the histological specimen, it's a little harder to see, but you can see this. Remember, you're looking at a dead kidney. So you're looking at this. There's a medulla right there. There's a papilla right there. I'm sorry, there's a pyramid right there, and a papilla would be right here. Here's another one. There's a papilla right there, and there's the minor calyces right in here. Feeds the major calyces, which will eventually feed the renal pelvis. There's the cortex on the outside of the kidney. Now, the blood supply is delivered via the renal arteries, and they deliver about 1/4 or 1,200 mils of cardiac output to kidneys each minute. So 1/4 of what comes out of the heart in each minute, remember that what comes out is going to be from the left ventricle, because it's going to be to the system. So to be out of the left ventricle would be a better way to say this. Whatever comes out of the left ventricle over every minute, 1/4 of that will actually feed to the kidneys proper. The arterial flow into the venous flow and the venous flow out of the kidneys follow these very similar paths. And the nerve supply is via this sympathetic fibers from the renal plexus. Now, obviously, the kidneys are very vascular because 1/4 of the left ventricular output goes to them. And so you'll see that the renal artery and the renal vein follow this track, and we're going to see that there's a process of feeding through the columns, the arteries and veins to the columns. What you'll see here is you'll see that renal cortex, and you'll see the cortical radial vein and the cortical radial artery that sort of feed this, and you're going to find that feeds off the arcuate artery, and then and so you're going to see that really supplies the blood. Provides the blood supply, what will become the filtering site, or the nephron units that we'll talk about in just a minute. So as we look at the blood supply, we're going to take a route through the kidneys. We start out at the aorta. That becomes the renal artery. That feeds to what's called the segmental artery, then the interlobal artery, and the arcuate artery, and then that fills all the cortical radial arteries that are in that cortex. That becomes the afferent arteriole, which will feed the glomerulus. That's where the filtering occurs, glomerular capillaries. That then leaves, becomes the efferent arterioles. They become the peritubular capillaries. Vasorectal, I'll explain that later. The cortical radial vein, arcuate vein, interlobal vein, renal vein, and finally the inferior vena cava. So that's really the path for the blood supply through the kidneys proper. Now it's the nephrons where really the filtering takes place, and they are the structural and function units that really form the urine or filter the blood. And we have about 1 million per kidney, plus or minus, so 2 million overall. And there are two main parts. There's a glomerulus, which is the capillaries, and this renal tubule, which surrounds it. And it's called the Bowman's capsule. I want to show that to you. We'll see this many times throughout this lecture. So here's our kidney. We've taken a small cut here. We go over here. And so what we're seeing is we're seeing the artery come in. We see what's called the afferent artery coming in. Afferent means coming to the glomerulus. It makes this little group of capillaries right here, and then it goes out being the efferent artery. What happens here at the renal corpuscle, as it's called, which includes the glomerulus, all those capillaries, and the cup around it, which is Bowman's capsule, is that it's all filtered. Everything is sort of filtered out. So basically what happens is it's all filtered out, and I always refer to this as kind of like when my grandparents used to have me clean the garage. And this will make sense in the end, I hope. So my grandparents, I lived on a ranch, and my grandmother had a separate garage for just her car. Now we had big garages for tractors and trucks and things, but my grandmother had her own garage. And she always kept her car in it, and it was to, she felt it was to keep the car, looking nice and that kind of thing. About twice a year, my grandmother would enlist me to clean the garage. And so I would go in the garage, and her idea of cleaning the garage was to remove everything and only have the car left in the garage. And so I would go in the garage, and I'd start to remove everything. And that's kind of what happens in Bowman's capsule. You remove everything, or everything that's going to be removed is pretty much done there. The bulk of that is removed there. And my grandmother was pretty happy for a bit, but then my grandfather would come along. And my grandfather was kind of a hoarder, not really a hoarder, but a saver. And what he would do is he would start to put things back in the garage. He'd say, well, I'm saving this axe handle just in case, or I'm doing this just in case. He'd go on and on. And so stuff would go back in the garage. And that's kind of what happens after Bowman's capsule. So we have Bowman's capsule here, and Bowman's capsule is where most of the things are filtered out. And then as we go down the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule, things are put back into the blood, and I'll explain that in a bit, how that works. So everything's filtered out, and then it's reabsorbed back in the blood. So my grandmother is at work, I always say, in the glomerulus, and my grandfather is at work in the proximal convoluted tubule, loop of Henle, and the distal convoluted tubule. So that's just kind of a way to understand how it plays. So there's our glomerulus again, and we can see that we can look at that. Now that's not the whole story. There's a whole bunch of other things that go along with that, but that's kind of a good start. So like I said, the renal corpuscle is that glomerulus, which is a tuft, if you will, of capillaries, as well as Bowman's capsule, that glomerular capsule. And then we have around that a fenestrated glomerular endothelium that makes that capsule, and it allows that filtrate to pass from the capillaries through it into the glomerular capsule. So we'll see that. Now, the renal tubule, the part where the filtering occurs, starts with that glomerular capsule, that Bowman's capsule, and there's a couple layers, and we've talked about parietal and visceral layers before, parietal being the outside layer and visceral being the inside layer. And what you're going to see is those two layers. And the inside layer, the parietal layer, or the outside layer, is really a simple squamous epithelium. And the visceral layer, the inside layer, is made up of these little podocytes. And podocytes are cells that have little feet-like extensions. And the extensions will terminate in the foot processes that cling to the basement membrane. Now this is something we've seen before in lots of different places. We saw it in the lungs and the alveoli and the capillaries. There's a capillary, there's a basement membrane, and then there's whatever it's attached to. And in this case, in the renal system, it's the capillary, it's the basement membrane, and it's these podocytes. So they are these podocytes extend, and the spaces between the feet, if you will, the podocytes, is where the filtrate will pass into the Bowman's capsule. So what you're going to see is there's that glomerular parietal layer. We said that squamous epithelium. And then we have these podocytes, and there's our capillary, a fenestrated capillary. There's our basement membrane, which is the glue that holds these two together, and there's our podocyte, and the urine is going to filter through these little slits between the feet of the podocyte. Podo means foot, and cyte means cell, so foot cell. Now that will feed into what's called the proximal convoluted tubule. That's at first, and convoluted just means where it moves back and forth. And the proximal convoluted tubule is lined with cuboidal cells with dense microvilli and large mitochondria, cuboidal epithelial cells. And if you go all the way back, we talked about tissue and epithelial tissue particularly. We said that cuboidal cells function by in reabsorption and secretion. So what you're seeing now is that coming to life. And these are confined to the cortex, if you will. There's our cuboidal cells. Here's our microvilli on there, and we're seeing this proximal convoluted tubule cells with that. The next part is that loop of Henle, the loop at the bottom of the nephron unit. Loop of Henle has a descending loop, and then it goes back up again, the ascending limbs. And the thin segment, that is the descending limb, is made of simple squamous epithelium, and it's very permeable to water. The thick segment, the ascending limb, is made up of cuboidal and columnar cells, and it is very unpermeable or apermeable to water. So water leaves from the descending segment, but it cannot leave from the ascending segment. We'll see a little more of that information later. There's our loop of Henle, our thin segment cells that we talked about. Now, the renal tubule then goes to distal convoluted tubule. It's made of cuboidal cells with very few microvilli. There's still some secretion and reabsorption, but more secretion than reabsorption. And again, it's confined to the cortex of the kidney. There's our distal convoluted tubule, very rare microvilli at the top, lots of mitochondria you can see down below. That, of course, leads to the collecting ducts, and they receive the filtrate from the many nephrons, and they fuse together to eventually deliver urine through the papillae, or the point of the pyramid in the medulla, and into the minor calyces, where the urine is now collected. Now, we look at collecting ducts, the cell types. There's intercalated cells. We have cuboidal cells with microvilli, which function in maintaining this acid-base balance of the body. We'll see that. And there's our principal cells, our intercalated cells, if you will, in the collecting ducts. They'll manage our bicarb excretion and secretion. And then we have principal cells that are cuboidal cells without microvilli. They help maintain the water balance and the salt balance. Let me translate water and Na and sodium. Here we see another look at a nice nephron unit. We've taken the same cut, and we're looking at where those cells are located along the track of this process. There's our afferent artery, there's our glomerulus, there's our efferent artery. Here we have our glomerulus, and there's our Bowman's capsule, renal corpuscle, including glomerulus, proximal convoluted tubule, descending loop of Henle, ascending loop of Henle, distal convoluted tubule, and then our collecting duct. Now, nephrons, we have cortical nephrons, and about 85% of the nephrons are cortical nephrons, and they're almost located entirely in that cortex of the kidney. And then we have juxtamedullary nephrons, and they are nephrons that where the loop of Henle actually deeply invades in the medulla, and they have these extensive thin segments, and they're really important in the production of concentrated urine, or pulling out all the water, if you will, out of the filtrate, and we'll see that in just a bit also. So this is the rest of the story. So let's look at this a little closer, and let's see here we have this nephron unit. We said that we have an afferent artery goes in. There's the glomerulus, and the efferent artery comes out. Well, the efferent artery, as we look back into the flow of blood, actually becomes this sort of this capillary net that surrounds this long loop of Henle. And if you think about it, let's think about this in an osmotic sort of way. At the glomerulus, the pressure of the blood, the hydrostatic pressure, pushes the fluid out into the collecting system. So now the blood that leaves this glomerulus through the efferent artery that's coming down is very concentrated. Now, the fluid, if you will, that's in the collecting ducts is very dilute because it took most of the water out. So if you remember osmosis, that's fluid moving from an area of low concentration to an area of high concentration. And that would just fit nicely with what's going to happen next. So that afferent artery, the efferent artery, I'm sorry, as it comes out, becomes what's called the vasa recta. It's a net of capillaries around this loop of Henle, and they are in intimate contact with this loop. It's not like they're just hanging around, but they're attached. And through the process of osmosis, elements are brought back, water specifically is brought back from this tube back into the bloodstream. Now, you filter out about 95 liters of fluid every day in your kidneys. Now, you don't pee 95 liters because that'd be a lot. Because what happens is 95 liters comes out here, and most of it, but about 1 liter, 94 liters, is absorbed back in here. Now, let's put that in perspective. 95 liters is really about 24 gallons. Okay, 24 gallons of water. You figure about 4 liters per gallon roughly. And so that's about 24 gallons of water. That'd be a lot. So what you're seeing here is you're seeing that's reabsorbed back into our bloodstream. Plus, can you imagine trying to keep up with that? You'd have to drink 4 gallons of water every day, so you'd pee 4 gallons out, not including what you'd use. Pretty crazy. So efferent artery comes out. It becomes the vasa recta, these capillaries that will surround these. You'll see this sort of peritubular capillaries, if you see here. And they'll surround that here and here, in the short one as well as the juxtamedullary nephron units, and they will reabsorb that fluid back in and some of the electrolytes also. So let's talk about the nephron capillary beds. We start out with the glomerulus. We have the afferent arteriole going into that glomerulus, Bohn's capsule becomes the glomerulus, and then it leaves as the efferent arteriole. And it's very specialized for filtration. The blood pressure is high because the hydrostatic pressure is driving the fluid from leaving the capillary, that fenestrated capillary, through the podocytes into the collecting system. So the afferent arterioles are smaller in diameter than efferent arterioles, and the arterioles are high resistant vessels. And this is to prevent our kidneys from suffering from any high pressure issues the body may have. It's a protective measure. Now, as the efferent arteriole leaves, it becomes these peritubular capillaries, or the vasa recta we talked about before. They do about a lot of the same things. These are low pressure, porous capillaries, and they are filled with blood that's very concentrated because we filtered all the fluid out, and like they rise to the efferent arterioles. They cling to the renal tubules in the cortex, as well as in the vasa recta in the juxtaglomerular units, and they empty back into the venules. And so at this point where we see these peritubular capillaries, we're seeing the osmotic properties pull that through oncotic pressure, is pulling that very dilute liquid from the urine back in the very concentrated area where the blood supply is in those capillaries. Now the vasa recta, they're long vessels that are parallel to the long loops of Henle and the juxtaglomerular nephrons, the ones that extend deep in the medullary part of the kidney, and they help to concentrate the urine. They're more involved with pulling that fluid or that water back. And we've already seen this slide, but again, you see these peritubular capillaries, and you see the vasa recta, which are pulling that fluid that was created by grandmother at work in the glomerulus, and my grandfather at work in the collecting system to return the fluid back to the bloodstream. Now, the vascular resistance to the microcirculation, there's a high resistance to the afferent and efferent arterioles, and it causes the blood pressure to decline from about 95 millimeters of mercury, which is pretty high, down to about 8 millimeters of mercury in the kidneys. And that, again, that's a defense mechanism or a survivor mechanism to keep the kidneys. Can you imagine what happened if 95 millimeters of mercury in the capillaries of the kidneys? It would just blow up. So that would be a bad thing, obviously. Now, resistance to the afferent arterioles, the ones going into glomeruli, they protect the glomeruli, like I said again, from fluctuations in the systemic blood pressure. And resistance to the efferent arterioles reinforces that high glomerular pressure. It reduces that hydrostatic pressure because we know that that's going to be less, which allows the oncotic pressure, the osmosis, to take over and return the fluid back into the bloodstream via the peritubular capillaries and that vasa rectus, excuse me. Now, the juxtaglomerular apparatus, there's one per nephron, and it's really important in regulation of filtrate formation and blood pressure. And it involves modified portions of the distal portion, the ascending limb of Henle, and the afferent, sometimes efferent arteriole. And we want to take a look at that. We have granular cells, which are juxtaglomerular or JG cells. They're in large, smooth muscle cells. They're arterial. They have secretory granules, which contain the renin. Remember the renin-angiotensin cycle. And they act as mechanical receptors that sense blood pressure. So what you're seeing here, well, look, I'm going to go forward and I'll come back. So there's this juxtaglomerular apparatus. I've got to say it correctly. There's the afferent arterioles, which are feeding the glomerulus. There's the efferent arterioles coming out, and there's this tube, if you will. There's that ascending loop of Henle right here, which is coming back up, and you see there's the macular densa cells, and then we see the extraglomerular mesangial cells, and we see the granular cells located right here. So this is the apparatus we're talking about, located in that ascending loop of Henle we talked about. So let's go back and just review this a bit. So there's those granular cells. They're large, smooth cells along the arterial. They have the secretory granules, and that's where our renin comes from those granular cells. The macula densa that I pointed to, it's a closely packed set of cells in the ascending limb, and it acts as chemoreceptors that sense sodium chloride, salt, content to filtrate. The extraglomerular mesangial cells, they are interconnected with the gap junctions. They may pass signals between the macula densa and the granular cells. So that apparatus, helps us sort of manage this process of controlling pressures as well as controlling maybe how our filtration occurs. So you can see that mechanism. Now the filtration membrane, it's a porous membrane between the blood and the capsular space. So this, they talk about we're back to Bowman's capsule, if you will. And it consists of these fenestrated endothelium of the glomerular capillaries. So it's fenestrated capillaries, so they have pretty open gaps. And the visceral membrane of the glomerular capsule, which contains those podocytes, and they're connected together with that basement membrane we talked about. It's fused basal lamina of the two layers. And just like we learned in the pulmonary system, they are connected together, sort of glued together via this basement membrane. Now here we see that, here's the apparatus, and we enlarge that. So here's our fenestrated capillary inside, and you see the podocytes on the outside, and we see that fluid then pours through this, is pulled through this, and then goes down that proximal convoluted tubule. So you're seeing the podocyte cell body, and there's the feet of the podocytes, and then the capillary, that fenestrated capillary, which is underneath. And remember, the afferent arterioles are higher pressure, so they are pushing the fluid out because the hydrostatic pressure is in charge here. The orthostatic pressure, the onconic pressure doesn't really play a part until it gets down to the peritubular capillaries. Now the filtration membrane allows passage of water to solutes smaller than most plasma proteins, but they prevent filtration of red blood cells. So the red blood cells stay in the arteries, the afferent arterioles, and negative charged basement membrane repels our large anions, such as the plasma proteins. So it keeps the larger plasma proteins in. So it doesn't take everything out, but it leaves some things in. And the slit diaphragms also help to repel other macromolecules. So it's really just taking out waters and solutes smaller than those plasma proteins. Now the glomerular mesangial cells we talked about before, they engulf and degrade the macromolecules, and they can contract to change the total surface area that's available for filtration. So they can change the rate of filtration based on blood pressure or concentration in the blood. There's our capillary, our fenestrated capillary. We have those large pores. There's our basement membrane. There's our feet of our podocytes, and there's the filtrate that's leaving through that. Now, the mechanism of the urine formation is the kidneys filter out the body's entire plasma volume 60 times per day. That's quite a lot. And the filtrate is really the blood plasma minus those proteins. That's what it really is. And the urine is really only 1% of that total filtrate. I think I said that before. And it also takes out our metabolic waste and the unneeded substance or the results of metabolism. Now, the mechanism of urine formation, we have glomerular filtration, and we've talked about that before, where my grandmother's at work is filtering out most everything. And then we have tubular reabsorption. That's where my grandfather's at work. He starts to put stuff back into the garage to return some glucose, and amino acids, and 99% of the water, and the salt, and other components of blood. Now that's the main process. there's a minor sort of a fine-tuning process called tubular secretion. And that is where, even at the point of the tubules, there will be a point where the blood is going to actually put some things into the filtrate, into the what will become urine. And it's the reverse of reabsorption, and it selectively adds things back to the urine. It's kind of the fine-tuning mechanism. You might say this is the big process, and then this is the fine-tuning mechanism. And we'll talk about how those things occur. So here we have, we're looking at glomerular filtration, tubular reabsorption, and tubular secretion. Tubular filtration is what happens in the glomerulus. It's where everything comes out. Tubular reabsorption is really what's happening if we look at 2 down here, and we're seeing tubular reabsorption where things are moving back from the urine back into the bloodstream via that osmotic process. Now there's also tubular secretion, that fine-tuning that occurs, and that's going to be what happens here. Once we get through the reabsorption process, it's kind of the process that says, okay, we need to even these amounts out or make these more precise, more or less, and right here. And so you see that effect. Now, glomerular filtration is a passive mechanical process, as we know, which is driven by the hydrostatic pressure in the glomerulus. The glomerulus is a very efficient filter because its filtration membrane is very permeable and has a large surface area, and the glomerular blood pressure is higher than other capillaries, about 55 millimeters of mercury. The molecules that are greater than 5 nanometers are not filtered like plasma proteins, and they function to maintain the colloidal osmotic pressure of the blood. Well, what does that mean? It means that we take the fluid, allows the fluid to stay out, but the osmotic pressure, the osmosis power of the blood rises because we take the fluid out. And we know that's going to be important. We get down to the peritubular capillary process and the vasorecta. The net filtration pressure is the pressure responsible for filtrate formation. It's about 10 millimeters of mercury difference in that area. The net filtration rate is determined by the glomerular hydrostatic pressure, the chief force, and the 2 opposing forces, the colloidal osmotic pressure of the glomerular blood and the capsular hydrostatic pressure. So the hydrostatic pressure of the afferent arteriole forces the fluid out, but it has 2 resistant pieces. That's the colloidal osmotic pressure of the blood that may be trying to pull it back, but it's a minor player. And then the pressure inside the capsule itself, which is also a minor player. So net filtration rate is this power minus these 2 powers to create this net result, which is about 10 millimeters of mercury in pressure. So here we see this pressure coming out from the glomerulus. There's the afferent arteriole, and there's the efferent arteriole. We see that the pressure is pushing out here. That's the glomerular blood pressure is about 55 millimeters of mercury. We have blood colloidal osmotic pressure, which is trying to pull it back in. That's about 30 millimeters of mercury. Then we have capsular hydrostatic pressure, which is pushing against all of this. And 15 plus 30 is 45, which gives you about that 10 millimeters of mercury pressure that we talked about. Now, the glomerular filtration rate, that's the volume of filtrate formed per minute by the kidneys, is about 120 to 125 mils a minute. And actually, that's a healthy kidney. If it's around 125, we know the kidneys are working well. And it's governed by and directly proportional to the total surface area that's available for filtration. We saw the mesangial cells sort of manage that, and the filtration membrane permeability, and the net filtration pressure. So we know that that plays an important part, that 10 millimeters of mercury that we talked about before. Now, the glomerular filtration rate is tightly controlled by 2 types of mechanisms. Our intrinsic controls, that's the renal autoregulation. That's what's acting within the kidneys. We know that's the mesangial cells. And then we have extrinsic controls, which are the nervous and endocrine mechanisms that help to maintain blood pressure, but they can very much affect kidney function, and we've talked about these before. The intrinsic controls, they maintain a nearly constant glomerular filtration rate when the mean arterial pressure is in its normal range, or in the range of 80 to 180, if you will, which is a little high end, 80 to 180. There are 2 types of renal autoregulation. There's the myogenic mechanism, if you will, and then there's the tubular glomerular feedback mechanism, which senses the changes in the juxtaglomerular apparatus we talked about and those mesangial cells. So we've already kind of referred to this before. Now, the myogenic mechanism is, as the blood pressure increases, they will perpetuate a constriction of those afferent arterioles, and that lowers the pressure in the glomerulus because it restricts the blood flow. It helps maintain a normal glomerular filtration rate, and it protects the glomeruli from damage from the high blood pressure. Now, this is not a perfect system. It can be overcome, and it happens in people who have malignant hypertension. Now, the opposite is true also. If there's a decreased blood pressure, it will dilate those afferent vessels and try to maximize the glomerular filtration rate so that we can keep blood flowing to the glomerulus. Now, the tubular glomerular feedback mechanism is a flow-dependent mechanism directed by the macula densa cells we talked about. If the GFR, the glomerular filtration rate, increases, the filter flow rate increases in the tubule. If the filtrate sodium chloride, salt concentration, will be high because of insufficient time for reabsorption. So think about, I always refer to this as the I Love Lucy phenomenon. If you ever watched the show I Love Lucy, there's one that you probably saw even on Nick at Night or one of those shows. And what happens is Lucy is working at a candy shop, and her job is to put candy into boxes, and the candy is coming down one piece at a time down this conveyor belt. And she starts to put the pieces in the box, and things are going fine until something happens with the machine, and the flow of candy gets very, very high, and lo and behold, she can't keep up, and she starts shoving candy in all sorts of places. Well, the thing to take from this as far as glomerular filtration rate is if the flow rate is very fast, a lot of things will get through and not be reabsorbed, so they'll go into the urine. If the glomerular filtration rate slows, what happens is that it allows the reabsorption to be more effective, and it is able to pick up more things. So if like the conveyor belt in the I Love Lucy metaphor, she can pick up more candy and put it into the boxes. If it goes too fast, she misses some of the candy. And so this filtrate of sodium chloride concentration will be high because it doesn't have enough time to reabsorb that. Now, the macula densa cells of the juxtaglomeric apparatus, they respond to an increased sodium chloride by releasing a vasoconstricting chemical that acts on the efferent arteriole and decreases the glomerular filtration rate. So if the sodium chloride is really high, it'll decrease the rate, which helps it to pull in more of that salt. The opposite occurs, the GFR decreases. If just the opposite happens, it moves a little faster. So the extrinsic controls, we have sympathetic nervous system under normal conditions at rest. The renal blood vessels are dilated. That's the way they work. The renal autoregulation mechanisms prevail. Remember, the sympathetic system only kicks in when it's trying to shunt blood to our arteries of our skeletal muscles and our heart and our lungs. That fight or flight mechanism. So it steals blood from our digestive system and our renal system. So when the sympathetic nervous system kicks in, it's going to obviously constrict these vessels, so it slows down that process. So under extreme stress or in the fight or flight mechanism, norepinephrine is released by the sympathetic nervous system, epinephrine is released by the adrenal medulla, and both cause constriction of the afferent arterioles, which inhibit filtration and trigger the release of renin in the renin-angiotensin system to retain fluid. The idea in this is the kidneys aren't getting enough blood, and they think that they need more fluid. We are shunting our blood to our arteries and veins of our heart and lungs and our skeletal muscles to outrun the bear that's chasing us through the woods in that fight or flight mechanism. So the renin-angiotensin mechanism is triggered when the granular cells of the juxtaglomeric apparatus release renin. Angiotensin is a plasma globulin. This should be renin, if you will, not resin. Renin changes that to an angiotensin 1, if you will. And then what happens is angiotensin converting enzyme, an ACE, if you will, converts angiotensin 1 to angiotensin 2. Now, this is important to know because ACE is going to change angiotensin 1 to angiotensin 2, and angiotensin 2 is a very strong vasoconstrictor, and it helps to retain fluid. Now, we actually have antihypertensive medications which are inhibitors of this. ACE inhibitors are called, and they're angiotensin converting enzyme inhibitors, ACEI. So this is a place that we work to inhibit that angiotensin response here to reduce high blood pressure. All a part of angiotensinogen, sorry, it's not angiotensin, angiotensinogen, which is addressed by this renin, which changes to angiotensin and on down the line. Now, the effects of angiotensin 2, it constricts the arterial surface smooth muscle, which causes the mean arterial pressure to rise or to raise our blood pressure. It also stimulates the absorption of sodium. Now, you know, wherever sodium goes, water follows, and so if it helps us forces the reabsorption of sodium from the renal tubules, from the renal tubules, proximal convoluted tubule and the the loop of Henle and the distal convoluted tubule, it'll promote reabsorption of sodium, then water will follow, and it triggers the adrenal cortex to release aldosterone, which does exactly the same thing. It also pulls sodium in. It also causes the water to follow, and it stimulates the hypothalamus to release ADH, and that also activates the thirst center, and the ADH is also going to pull sodium over and have water follow. So it's kind of a threefold process. This constricts the efferent arterioles, decreases the peritubular capillary hydrostatic pressure, and it promotes more fluid coming in through that osmotic process where the fluid is leaving the tubules, coming back in the bloodstream via those peritubular capillaries or through the vasa recta. This causes the glomerular mesangial cells to contract, decreases the surface area available for filtration, and its goal is to pull all the fluid back in. Now, this renin-angiotensin mechanism, the triggers for that renin released by the granular cells is a reduced stretch of the granular cells. The mean arterial pressure falls below 80 millimeters of mercury. Stimulation of the granular cells by activated macula densa cells, or direct stimulation of the granular cells via a beta adrenergic receptors by the renal nerves. So all of these are triggers for that. And this is a nice, large sort of track you can look through in your book, which follows everything we just talked about before, and I'm not going to go through it again. Now, other factors that affect your glomerular filtration rate include what's called prostaglandin E2. It's a vasodilator that counteracts vasoconstriction by norepinephrine and angiotensin II, and its primary goal is to prevent renal damage when the peripheral resistance is increased. So it's a leveler. It's a sort of a way to sort of keep things from going too extreme. Other factors include intrarenal angiotensin II. It reinforces the effect of the hormonal angiotensin II, and then adenosine, which is a vasoconstrictor of the renal vasculature. So you can see that all these mechanisms, extrinsic and intrinsic, really play a part and work together. Now, we've left the glomerulus, and now we're going down to the tubular area, and we're talking about tubular reabsorption. This is my grandfather at work, and that is a selective transepithelial process where the organic nutrients are reabsorbed back to the blood. Water and ion reabsorption are hormonally regulated, and includes an active and passive process. There are two routes, a transcellular and a pericellular process. The transcellular route is the luminal membranes of the tubule cells and the cytosol, in the cytosol of the tubule cells. The basolateral membrane of the tubular cells and endothelium of the peritubular capillaries. So we also have the pericellular route that's between the cells, and it's limited to the water movement and reabsorption of calcium, magnesium, potassium, and some sodium in that proximal convoluted tubule where the tight junctions are leaky. And I love this statement. Tight junctions are leaky. They're either tight junctions or not, but in this case, they're not so tight. They're sort of half tight. Here we see that effect. We see the fluid that's moving in the pericellular area and what's going through the cellular area. And you can see that we see water and calcium and those other minerals that come through that, those other ions, and then we have the luminal membrane and those other things that are going through. Now, sodium reabsorption, that's the most abundant cation in the filtrate, obviously, because of the blood, 280 milliosmoles of the 300 milliosmoles in the blood are really sodium. It's the primary active transport out of the tubule cell by the sodium, potassium, ATPase in the basolateral membrane, so it's what it's for. And sodium passes in through the luminal membrane by the secondary active transport to facilitate diffusion mechanisms. So what this really is happening is that there's a facilitate diffusion and there's an active transport part that plays in this, but there's also a passive part. And that's at low hydrostatic pressure and high osmotic pressures in the peritubular capillaries promote the water and solutes from being brought back in to the blood supply. And I've said that several times now in several ways. There's also reabsorption of nutrients, water, and ions. Sodium reabsorption provides the energy through that secondary transport. Remember, sodium comes through and pulls other things with it. It means for reabsorbing most other substances. So sodium is pulled through, and other things are pulled through as secondary active transport. Organic nutrients are reabsorbed by the secondary active transport, and the transport maximum, if you will, reflects the number of carriers in the renal tubules that are available. And they can become saturated, so that the sodium can only pull so many of these through those carriers, if you will. So when they get saturated, the excess is then excreted out the urine. So you know that if there's a lot of that, only so much will come back in, and that's a leveling mechanism to ensure we don't reabsorb everything back in. Now, water is reabsorbed by osmosis, and we already know that, obligatory water reabsorption, and it's aided by these water-filled pores called aquaporins, which we've talked about way back before. Cations and fat-soluble substances follow through by just clear diffusion. They just diffuse right through. The cations following cations following through secondary transport in some cases, and the water following also. And this talks about that also. We kind of talked through this, so I'm not going to walk through these steps right now. This is in your book in Figure 25.14. Now, we look at reabsorption of the proximal convoluted tubule, and that's the site of most of our reabsorption. 65% of the sodium and subsequently the water is reabsorbed at this level. All our nutrients, the ions and small proteins, are reabsorbed at the proximal convoluted tubule, the tubule between Bowman's capsule and the loop of Henle. Now, the loop of Henle, the descending loop is very open to water movement, and the ascending loop is only open to sodium, potassium, and chloride, but no water movement. And we'll talk more about that in just a bit. The distal convoluted tubule, the tubule after loop of Henle and the collecting duct, reabsorption is hormonally regulated here. Calcium is reabsorbed through the parathyroid hormone. Water is reabsorbed here through the antidiuretic hormone. And sodium can be reabsorbed here, but only through aldosterone or the atrial natriuretic peptide that the heart produces when it's injured. Now, the mechanism to aldosterone is it targets the collecting ducts to principal cells in the distal convoluted tubule, and it promotes the synthesis of luminal sodium and potassium channels. And it promotes the synthesis of basolateral sodium and potassium ATPases. Now, the reabsorption is in reverse. The potassium, the hydrogen, and the NH4, if you will, creatinine, organic acids, move from the peritubular capsules, the tubular cells, into the filtrate. So this is a secretion process, which is that reabsorption reverse. That's what happens, is that leveling sort of way. It disposes of substances that are bound to plasma proteins, and it sort of levels these other elements. So we see that it kind of manages our acid-base balance a bit here through hydrogen. It manages our potassium, NH4, creatinine, organic acids. And we'll see that that plays a part in that. Now, secretion, which is the movement of blood from the blood into the urine at the end, eliminates undesirable substances that have been passively reabsorbed, like urea, uric acid, and it rids the body of excess potassium, fine-tunes the potassium. It controls the blood pH by altering the amount of hydrogen and bicarb that's pushed in the urine. Remember, hydrogen or bicarb is the thing that kidneys produce in our metabolic acid or metabolic acidosis. It helps to manage that acidosis. Now, the regulation of urine concentration and volume is through osmolality, the number of solute particles in 1 kilogram of water, and it reflects ability to cause osmosis. So osmosis really helps to manage a lot of our concentration. The osmolality of body fluids expressed in milliosmoles, and the kidneys maintain an osmolality in plasma of about 300 milliosmoles. Remember that 280 of that is really sodium. So they manage the sodium primarily. So most of the job is managing sodium by the kidneys. It uses the countercurrent mechanism. That is, we absorb all the sodium at the Bowman's capsule, at the glomerulus, and we selectively put it back in the blood through this countercurrent mechanism. Now, the countercurrent mechanism that it's primarily effective is going to be in the loop of Henle. And this loop of Henle occurs when the fluid flows in the opposite direction of the two adjacent segments of the same loop. And the filtrate flow in the loop of Henle is a countercurrent multiplier, and the blood flow in the vasal rectus. So let's take a look at that a bit. And I want to go forward. We'll see that let's go back just a second. I'm sorry, I went too far. So the role of the countercurrent mechanism helps establish and maintain that osmotic gradient, about 300 milliosmoles in the blood. It can go up to 1200 milliosmoles in the renal cortex through the medulla, and allows the kidney to vary that urine concentration through that process of controlling osmosis. Now, the descending loop is freely permeable to water, which passes out the filtrate in the hyperosmotic medullary interstitial fluid, and the filtrate osmolality can increase up to 1200 milliosmoles because guess what? We've taken all the water out, so it's far more concentrated. The ascending loop, on the other hand, is impermeable to water. It's selectively permeable solutes, primarily sodium and chloride, which are passively reabsorbed here, and they actively reabsorb the thick segment. They filtrate osmolality, will go down from that 1200 down to a mere 100, because guess what? We're now pushing the solutes back out. Now I want to show you this because you have to think of this not as a photograph, but as a video. So fluid is moving through here. Remember I told you wherever sodium goes, water follows, and that makes this not so intuitive. So water is taken out of the filtrate, put back in the bloodstream here, and sodium chloride is taken here. So what happens is that this is dictated by the concentration of the water. So the amount of sodium that's being put through is dictated by how much water is taken out. It's sort of a backwards mechanism. It's still this, wherever sodium goes, water follows, but what happens here is the sodium here is going to dictate how much water is taken out here. So if sodium is pushed back in the blood, more water will be taken out here. If sodium is kept in the urine, then more water will be kept in the blood, or in the tubule and kept in the urine. So even though this happens before this, it's so quick it's actually not within the same millisecond, if you will. So even though water is pulled here, sodium is pulled here, this is dictating the water movement here based on the concentration of sodium here. I just want to sort of get that in your brain. So all water comes out of the proximal convoluted tubule, no water comes out of the distal convoluted tubule, but this is the place where we tend to see electrolytes, and primarily sodium, because the 280 milliosmoles of the 300 milliosmoles of blood is really sodium. Now we also remove urea between the collecting ducts and the loop of Henle, and urea is recycled and secreted into filtrate by the facilitated diffusion in the ascending thin segment, and it's reabsorbed by the facilitated diffusion in the collecting ducts deep in the medulla. And it contributes to the high osmolality in the medulla, so it contributes to that being up to that 1200 we talked about. Now we talked about the vasorecta before, but they also play a part just like the peritubular capillaries. They maintain the osmotic gradient in those nephron units where the loop of Henle goes deep in the medulla. They deliver blood to the medullary tissues. They protect the medullary osmotic gradient by preventing rapid removal of salt and by removing reabsorbed water. So they are more effective in that process, and you see that process in that loop, in that deep juxtaglomerular nephron unit, that juxtaglomerular loop of Henle. And so you see sodium and water is being pushed in. You see here that we'll see a movement that's going to occur a little differently in the vasorecta. Now the formation of dilute urine, filtrate is diluted in the ascending loop of Henle. In the absence of ADH, so basically in ADH, the antidiuretic hormone, the dilute filtrate, will continue out the renal pelvis as diluted urine. So ADH, if you have no ADH, antidiuretic hormone, which if you have the hormone, it helps to retain fluids. It keeps them into the bloodstream. If you have no ADH, it pushes it out. And I always remind you that ADH, there's a ADH inhibitor called alcohol. So if you drink alcohol, one of the first things you have to do is go to the bathroom. So if you have so you know that ADH is going to retain fluids. Alcohol is a ADH blocker, so it allows the fluid to move out no matter what the ADH is, and you get dehydrated when you drink too much over a period of time. So in the absence of ADH, the urine stays dilute. The filtrate continues into the renal pelvis as dilute urine. Sodium and other ions may be selectively removed in the distal convoluted tubule and collecting duct, decreasing the osmolality to as low as 50 milliosmoles. Now, and we'll see that effect here, and through that process of the descending loop, taking out our water and all of that. We've already talked about that. Now, the formation of concentrated urine depends on the medullary osmotic gradient in ADH. If ADH is created, it triggers the reabsorption of water in the collecting ducts, and through facultative water reabsorption, thanks to ADH, it takes out 99% of the water in the filtrate, and so the urine becomes more concentrated. It has less water in it. And we see that effect here, where eventually what you're going to see in that collecting duct is less water. Now diuretics, they work by moving sodium basically, and they can osmotic diuretics, the substances aren't reabsorbed. In other words, they keep sodium in the urine, and so they keep the sodium in the urine, and so if the sodium stays in the urine, so does the water that follows sodium, and it doesn't get back to the bloodstream, which lowers the blood pressure. It also will help glucose, by the way, because sodium, remember, through secondary transport pulls that glucose with it. Now, we have ADH inhibitors such as alcohol, and we know that allows the urine to flow out, and substances that inhibit sodium reabsorption, obligatory water reabsorption, work like caffeine, which can make you urinate also, and there are several drugs that can make you work like diuretics. So in the proximal convoluted tubule, 65% of the filtrate volume is reabsorbed. Sodium, glucose, amino acids, and other nutrients are actively transported. Water-emitting ions follow passively through here. Hydrogen and NH4 secretion and bicarb reabsorption occurs to maintain a blood pH, or the level of free hydrogen. We'll see that we saw that we'll see that in chapter 26. And some drugs are secreted, if you will. Some drugs are secreted into this area too. So we see those different areas, and we see what happens in those different areas through that. There's A. In the B area, so now we're seeing the B area here, we're seeing the ascending loop of Henle. We're seeing it's freely permeable to water. It's not permeable to sodium chloride. Its filtrate becomes increasingly concentrated as the water leaves the osmosis. So that's what happens there. In the ascending loop of Henle, it's impermeable to water, like we've talked about before. It's permeable to sodium chloride. The filtrate becomes increasingly dilute as water is reabsorbed here. At the D level, that's here in the distal convoluted tubule, sodium reabsorption is regulated by aldosterone, calcium reabsorption is regulated by the parathyroid hormone, and chloride is co-transported with sodium at this point. And then finally, what occurs in the collecting ducts here is water reabsorption to the aquaporins is regulated by ADH. Sodium reabsorption, potassium secretion regulated by aldosterone. Hydrogen and bicarb reabsorption or secretion to maintain blood pH occurs here. And the urea redistribution is increased by ADH. So you're seeing the different things that happen at each of these sites as we go through. Now, the way we look at kidney function is we do that by looking at renal clearance. And that's the volume of plasma that is cleared of a given substance at a given time. And renal clearance tests are used to determine what our glomerular filtration rate is, and detect if we have any glomerular damage, and they follow the progress of renal disease or renal failure. And what we do is we have renal clearance, that's the mils per minute, and that equals the concentration of milligrams per mil of the substance in the urine. The V is the flow of the urine formation, and those are multiplied against each other over the concentration of the same substance in the plasma. And the thing we do is what's called the creatinine clearance test. So the substance they're talking about is creatinine. We will take a blood sample and look at the amount of creatinine in the blood. We'll take the concentration of the creatinine in the urine and in milligrams per mil, and then we'll look, and that'll tell us the flow rate eventually of what's going to be the renal clearance. That tells us how much the flow is. So, not how much the flow is, it'll tell us the renal clearance is. The flow rate is going to be how much urine we produce. And in a renal clearance, what they do is they have you collect a 24-hour urine. They have you get a big jug and you collect all your urine. And that's taken to the lab. And the lab measures the total volume, which gives us your flow rate of mils per minute, because it's over 24 hours, so it's broken down to minutes. And then what they do is they take a sample, they mix up the urine so it's homogeneous. They take out a sample and they measure the amount of creatinine that's in every mil, the amount of milligrams in every mil. And so they already know U and V. And then they take the concentration of the blood and they do this calculation. And that tells them what the renal clearance is going to be. Now, for any substance freely filtered and either reabsorbed or secreted by the kidneys, like insulin or creatinine, the GFR should be around 125 mils per minute. And I mentioned that before we were looking at the glomerular filtration rate. If it's below 125 mils per minute, the substance is reabsorbed. If the renal clearance is 0, the substance is completely reabsorbed. The renal clearance is greater than 125, the substance is secreted. Most drug metabolites really fall into that line. Now, if the glomerular filtration rate shows 125 mils per minute or greater, then the kidneys are functioning pretty properly. If it falls below that, then we know we have kidney injury or failure. Now, when we look at urine, We have to look at urine based on the color and transparency. It's usually clear. It's pale to deep yellow due to urochrome. That's the thing that colors it. Drugs, vitamin supplements, and diet can alter that color. And a cloudy urine may indicate the urinary tract infection, actually, or a different smell, if you will. That odor is usually slightly aromatic when fresh. Develops this ammonia odor upon standing because that's a metabolic byproduct of bacteria. And it may be altered by some drugs and vegetables. The pH is slightly acidic in most cases, around 6, with a range of 4.5 to 8. Diet, prolonged vomiting, or urinary tract infections, or metabolic acidosis can alter the pH. Now, specific gravity, if you look at the specific gravity of water, it's considered 1. That is, it has a value of 1. When you add things to the water, the specific gravity becomes higher because you have more stuff in it. It weighs more, so to speak. And so the specific gravity of urine can be 1.001, very dilute urine, to more concentrated urine, which is 1.035. And we actually do a gravitometer. We take a small drop of the specimen, and we can look at the gravity of that urine with the gravitometer. Now, 95% of the urine is water and only 5% is solutes. It has the nitrogenous waste, the leftovers of our metabolism, like urea, uric acid, and creatinine. And it has other normal solutes: sodium, potassium, phosphate, sulfate, calcium, magnesium, and bicarb. It has abnormally high concentrations of any of these is kind of a pathology. It means something is going wrong. Now we looked at the kidneys. Now we're going to look at the next organ down. Those are your ureters. They're the tubes that run from the kidneys, from the renal pelvis down to the bladder. They convey urine for the kidneys, the bladder, and they are retroperitoneal also. They enter the base of the bladder through the posterior wall, and they are inferior and posterior in their insertion in the bladder. As bladder pressure increases, the distal ends of the ureters close, which is to help backflow the urine back to the kidneys. You don't want that to occur. That's kind of a problem. So the ureters have 3 layers of the wall of the ureter, and fluid or urine moves down the ureters through a peristalsis. People think it's gravity, but it's really peristalsis. There's really a peristaltic effect that moves the urine. If that weren't true, then when you lay down at night, your urine would just lay there. It wouldn't move down the bladder. So it's 3 layers. Its lining is transitional epithelium. It has a smooth muscle muscularis, which provides that contraction in response to stretch, so it gives us that peristalsis. And it has outer adventitia, the fibrous connective tissue, because remember, they're retroperitoneal. This is looking at the ureter. It's a collapsible membrane. You can see the adventitia outer layer. You see the circular and longitudinal layer of muscle, which gives you the peristaltic effect. And then there's the inside layer of epithelium, and there's the lumen. Now, you can get renal stones, renal calculi. Those are kidney stones which form the renal pelvis, and they're usually crystallized calcium, magnesium, or uric acid salts. It's interesting because most kidney stones are really it's interesting, they may be calcium on the outside, but most often they're started with uric acid salts. Isn't that interesting, uric acid crystals? So if you take almost any stone, kidney stone, and you cut it in half, the center, the nidus it's called, is really a uric acid crystal. Now, larger kidney stones can actually block the ureter, and because you're trying to go through peristalsis to move them into the bladder, that causes pressure and pain in the kidneys, and that's called renal colic. Now, kidney stones can be due to a chronic bacterial infection of the kidney, or urine retention, or too much calcium in the blood, or the urine is too alkaline. And so often what they'll have you do is if somebody has a chronic renal infection, so they have chronic renal stones, they'll have you acidify your urine by drinking acid juices, for instance, like grapefruit juice and all the other kinds of juices. You can have cranberry juice. Now, the urinary bladder is where the ureter is inserted to. It's a muscular sac for the temporary storage of urine. It's also retroperitoneal on the pelvic floor posterior to the pubic symphysis. In males, the prostate gland will surround the opening of the ureter coming out. In the females, it's anterior to the vagina and the uterus. And I'll say the vaginal vault and the uterus, and we'll look at that in just a moment. Now, it has a muscle inside called the trigone, like a triangle. It's through like triangular area. It's outlined by the openings of the ureters and the urethra. And if you have an infection in the bladder that is persistent, this is where the infection tends to be located. Now the layers of the bladder, it has the transitional epithelial mucosa on the outside. It has the muscle layers, the thick detrusor muscles, which help to contract to force the urine out when you're trying to urinate. And it has the it has a transitional epithelium mucosa inside, mucosa, the thick detrusor muscles on the outside, and there's adventitia on the far outside, the connective tissue, could be peritoneum or the superior surface, or the peritoneum is on the superior surface of the bladder only, the rest is is retroperitoneal. Now the urinary bladder collapses when it's empty, and the inside layer, if you will, has these roughed out areas they call rugae, these little roughened areas, unlike the stomach. It expands and rises superiorly during filling without significant rise in internal pressures. The pressure doesn't really increase. So let's look at a bladder here. We're going to see we see the inside rugae, and we see that urethral, that mucosa that's inside, and yes, it does produce mucus. It's a mucosal surface, and so we'll see it's transitional as well as columnar epithelium with goblet cells. The next layer we have is the muscle layer, and that's they see the detrusor muscle, which will help contract that bladder, and then the outside, that adventitia on the outside. Now, what you'll see here is you'll see the internal-external sphincter. The internal sphincter is that autonomic sphincter, and the internal sphincter is smooth muscle. That means it's directed by the autonomic system. It is involuntary. The external sphincters are skeletal muscle, and they are voluntary. So the internal sphincter, when the bladder gets enlarged, it gets full, it just would open up and let urine come out whenever or wherever. It's the external sphincter that helps us control the timing of that, so we can pee in the right times in the right places, and we don't, embarrass ourselves. And so this is the part that's going to manage that. We have those two sphincters we've talked about before. The ureters, as they come down, I want to show you that they're actually inserted in the posterior part as trigone, and they're inserted inferior and posteriorly. They come in underneath, so to speak, of the bladder. And this is we'll see this in the female primarily. In the urethra, it's a muscular tube, the tube that leaves the bladder. It's lined with epithelium. It's mostly pseudostratified columnar epithelium, except the transitional epithelium near the bladder and stratified squamous epithelium near the tip. So you're going to see columnar epithelium pseudostratified, and so there is a mucus production that occurs inside the bladder as well as part of the urethra. Now we have sphincters. I talked about the internal-external sphincter. That internal sphincter is involuntary. It has smooth muscle at the bladder-urethra junction, and it contracts to open whenever the autonomic system says so. The external urethral sphincter is skeletal muscle. It's voluntary, and it surrounds the urethra as it passes the pelvic floor, which gives us our control, so we have control over when that happens. Now the female urethra is about 3 to 4 centimeters in length, and if you remember that an inch is 2.54 centimeters. It's about an inch and a quarter, if you will. It's a tightly bound the anterior vaginal wall, and that's important also because the female urethra actually runs along the anterior vaginal wall. So when you're trying to do a catheterization of the urethra, urinary catheterization, you have to be very careful because you actually perforate that, especially in older women, because that tissue becomes more friable. It doesn't have the strength, and it's not as robust as it was before, and they can actually perforate, so you put the catheter into the vaginal vault. Not a good thing. And so the external urethral orifice is anterior to the vaginal opening and posterior to the clitoris or clitoris. Both those terms are correct. And we've already seen this process in the female. Now, in the male urethra, it carries semen and urine both. It serves both purposes through the same architecture. And there's 3 parts of the male urethra. There's the part that's in the prostate. That's the prostatic urethra. It's about an inch long. There's the membranous urethra. It's a little less than an inch long, passes through the urogenital diaphragm. And there's the spongy urethra. It's about 15 centimeters long, which passes through the penis and opens via that external urethral orifice. And we'll see there's the bladder of the male here, and there's the urethra opening up, and there's our prostate gland we'll see here. It actually surrounds the urethra. When males get benign prostatic hypertrophy, which means the bladder enlarges, it starts to impress upon this little area here. It starts to push in, and it can make urinating harder, and you'll hear that here. We have medications that help to do that, or sometimes we have to go in and take out some of that prostate, if you will. Cancer of the prostate will also affect this because it'll put pressure on this and impede, if you will, on that. So there's the prostate. There's the urogenital diaphragm, and we'll look at this later when we look at the reproductive system, but there's the we'll see here the external, these are Cowper's glands, if you will. They're the bulbourethral glands, and they're located on either side right here. And so there's our prosthetic urethra. There's our external urethra. We see our the small part of your urethra here until it becomes a spongy urethra. We see that, and this is the membranous urethra. I'm sorry, and the spongy urethra that's really part of that tract of the penis, and urine will go flow through this, as does semen, if you will, in the male. So we talk about micturition. That's really peeing, urinating, voiding. All those terms are accurate, and there's three simultaneous events that occur. There's contraction of the detrusor muscles by the autonomic nervous system, and there's opening of the internal urethral sphincter by the autonomic nervous system. Now, those are somewhat automatic. The opening of the external urethral sphincter is by the somatic nervous system or the voluntary nervous system. And this is how we control the time and place of our urination. Now there's reflexive urination, that's urination in infants, and that's distension of the bladder activates the stretch receptors. There's excitation of the parasympathetic neurons in the reflex center in the sacral region of the spinal cord. There's contraction of the detrusor muscles. There's contraction and opening of the internal sphincter. And there's inhibition of the somatic pathways of the external sphincter, allowing it to relax. Now, infants and young toddlers really don't gain full control of that external sphincter until they're about 2 years old, between 18 months and 2 years old. And so when you're talking about potty training, that becomes more effective between 18 months and 2 years, and not very effective prior to that time. Now, micturiation of pontine control centers mature between the ages of 2 and 3. The pontine storage center inhibits micturiation, inhibits the parasympathetic pathways, and excites the sympathetic and somatic afferent pathways. So between ages about 18 months to 2 years, really, we start to get control. The pontine micturiation center promotes micturiation. It excites the parasympathetic pathways, inhibits the sympathetic and somatic efferent pathways. So that's how we sort of manage that process, and we gain control through that training process. And this is the table 25.22 in your book. It sort of takes you through all of that, the things we just discussed. So we've looked at the renal system. We've talked about the function and physiology of the bladder. We've talked about the ureters, the bladder, and the urethra, excuse me, as we are moving through. Thank you very much.